Hello, Lekha. Can you hear me? Or else you can write in the chat box like if audio is not working properly. And Aditi joined today. You can see my screen, right? Yeah. I think I think you both were not able to join last week. Last week we did had a bit of introductory part of proteomics, whatever it was during the last week. This week it is majorly focused on one of the gel-based proteomics technique, which is two-dimensional gel electrophoresis. So we'll be just talking about one specific method, that is 2D gel electrophoresis. How exactly we can use that, how exactly uh, we can perform that specific experiment, and what all applications that specific experiment can be used for. We'll be dealing with all of those in today's lecture. I'll just see if I have started recording. Yes. I hope you all got the uh, previous week's PPTs as well as PDF documents as well as the videos. I have uploaded it, both the videos. Like you can, I think you all have access to the specific file wherein you can find the link for both the videos. Like if you want to revise them, and this is the specific folder which will help you to find the assignments basically. So I have added the not like assignments, the PPT which I'm currently like I generally share during our uh, this problem solving sessions. So I've uploaded PDFs as well as PPTs, and there are a few questions as well. I've added few of the extra questions, and these assignment solutions I have added, which are for previous year's assignments, whatever were there during the previous year course. I just added those questions as well as those assignment solutions, which will help you to uh, like deal with certain questions basically. So you can go through those links, you can go through those assignment questions. We also discuss those questions in our sessions as well. Still, if you all have any kind of doubts, we can talk about it in this class as well. So let's start today's lecture. So yeah, welcome to the proteins and gel-based proteomics course, which is an NPTEL course coding BT78. So in this week, this is week 3. We already had 2 week courses. Some of you were there, I think, during the first week, and some of like few of them were present in week 2 as well. So this in week three's session, sir had 5 lectures in this specific lecture, and I've divided them in like similar to the 5 lectures in 5 parts. So in the first lecture of week 3, sir majorly talked about uh, two-dimensional electrophoresis, but the initial step, what do we do with the so as it is two-dimensional, the first step is isoelectric focusing, second step is SDS page. So how exactly isoelectric focusing steps are done, like what is rehydration, how then we will perform the experiment, then we will do equilibration, etc. Then that was so part one is about the first dimension majorly. Then part two is about the second dimension, which is the SDS page running. And then once those two experiments are done, how exactly staining is done, how exactly destaining can be done. So there are different methods or different techniques available for your staining, which will help you, which basically tells us about different sensitivity of those uh, prediction basically. So once the gel is run on the like proteins are run on the SDS page gel. We need to in order to visualize them, we need to stain them, and that staining are of different types, which helps you. So different stains has different sensitivity. Some stain can detect even nanogram level of the protein. Some can detect even lesser than that. Well, some can detect only microgram levels. So depending upon your need, and accordingly the cost will also differ. The more the sensitivity, more is the cost. So accordingly, those different types of staining techniques are available. We can use them. Then once the staining is done, now we can visualize the gel. Once visualization is available, we can try and analyze the gel, like which proteins are getting overexpressed, which are proteins are getting down regulated, or some are which are getting up regulated. All of those things we can analyze using softwares as well as a bit visually as well. Once that is done, like Sir talked about different application, different case studies. The Sir talked about, and of like how this specific two-dimensional electrophoresis can be used. Similarly, after that, Sir talked about different challenges which can be used, or which are different challenges which people face in terms of performing this specific experiment, and that was in the part 5. So all this we'll be talking about in this lecture. So what I'll be doing is I'll be just going through all those specific parts what sir has tried to explain in his 5 lectures of week 3. I'll just revise all of those stuffs, and after that we'll go through certain questions. And if you all are having any kind of doubts in any of the part, may it be week 1, may it be week 2 or week 3, or any questions which you want to ask, we can discuss on that. And yeah, that's how we'll be proceeding with this lecture in today's class. I hope you all are getting it. Yes? Yeah. Okay. So let's start. So it's easy, as we are talking about only one specific experiment or one specific technique by which we can do proteomics, gel-based proteomic analysis. So in the first lecture, Sir talked about rehydration, isoelectric focusing, and equilibration. These are the topics he discussed. So what's the workflow? So first dimension is isoelectric focusing. Once that experiment is run, you will be doing equilibration step of that specific strip itself. Once it is getting equilibrated with certain buffers, then you will add that strip into STS page and run it vertically. Uh, then once that is done, you will be staining the gel, you will be doing the analysis, you can do spot picking, then that gel specific bands can be cut out and can be done enzymatic digestion, and we can further do mass spec analysis as well. So there are two analyses. You can initially do image analysis also, just to understand which proteins are getting upregulated, which are getting downregulated. We can do all of those stuffs. Then if affecting, like basically you will be having a diseased one and a control patient, and you will try and find the difference between them. Then mass spec will actually will allow you to have much more accurate results than that of image analysis. That's the reason we can further, if you want to do further analysis, then mass spec is one of the best technique to understand, like study proteomics basically. So why do we do 2D gel electrophoresis? What are the major objectives? So in terms of doing 2D gel electrophoresis, what do we do is the major aims or objective is to study global and differential protein expression and to resolve proteins from a complex mixture. So as we, like, you can use blood sample or you can use cell culture sample. Like for example, some cells are mutated and some cells are not. They will kind of produce different set of proteins. The normal wild type will produce different set of proteins. Like there can be a number of different example. And once you lyse those specific cells or extract specific proteins, now it will have a complex mixture. The 2D gel electrophoresis will help you to resolve them. Similarly, you can study isoforms and post-translation modifications as well. So because of the post-translation modification, like once the protein is formed, post-translation modification indicates glycosylation, phosphorylation, methylation, etc. Whatever. So if it is getting post-translated, it will have different nature altogether or different function. It will be more functional as compared to without post-translation of that protein. So you can accordingly study that as well in this technique, and as well as visual analysis of protein. As I just said, in terms of image analysis, you can do a bit of visual analysis as well by comparing the results between the wild type or the control with the disease or a mutated strain, whatever. Yeah. So just to give an overview of each and just to say in one or two liners what is 2D gel electrophoresis. So basically whatever your protein sample is, you will initially run the protein sample from in a pH strip, pH gradient strip kind of a stuff, which is called as IPG strip, isoelectric focusing strip, and then this will basically try and move the proteins in one specific direction from lower pH to higher pH, and basically it will the protein movement here will majorly depend upon only one thing, that is isoelectric point. So what is isoelectric point, if anybody can answer? Yes, what is isoelectric point? Can anyone answer? Either Lekha or Aditi. Isoelectric point can also be called as PI basically. Yeah. If people don't know what exactly isoelectric point is, so if not, then I'll answer. Separating proteins in the gel by where they are neutral by pH. Okay. Yeah. So this specific sentence or whatever you're trying to say, you are trying to define isoelectric focusing technique. I'm not asking you the question what is isoelectric focusing. I'm asking what is PI or what is isoelectric point. Yes. pH at which the protein charge becomes zero. Yes, exactly. That specific pH with no charge. Uh, Lekha, you know the concept. It feels like the way you are answering, but your sentence framing is somewhere not accurate enough to give the answer. So, if you want to define PI, it is a pH at which proteins are neither positive, neither negative, or basically it is a pH at which proteins are having no charge. That's the PI. Yeah. So for example, there was a complex mixture. Here there are one, two, three, four, five proteins. Maybe you can say the one which are having a pH around three, like PI around three, PI around five, PI around six, PI around eight, and around 11 or something. So depending upon the different PI, they are getting separated. So as there will be no charge at one certain point, they will be unable to move ahead towards negative charge. And after that you will take this strip and keep it in a horizontal direction, and once that is kept, you need to have an SDS page gel, and these are the proteins which are already loaded. You'll just have to load the ladder as such. Here there is no ladder image though, but you'll have to load ladder, and then you will be separating on the basis of molecular weight. Here the separation is on the basis of molecular weight, and here the separation is on the basis of isoelectric point. So this is what in a overall broad picture what 2D gel electrophoresis is. So once this is separated, then you will be doing staining, destaining, image analysis, mass spec, etc. So this is the experimental part. Then later it will be the more analysis part. So what is isoelectric focusing? It is protein separation according to isoelectric point. It basically proteins are introduced into a immobilized immobilized pH gradient, and electric field is applied in which proteins migrates according to its charge. So charge indicating the positive or negative charge, and then proteins are migrated in the presence of electric field, and as soon as they reach a specific pH, so there is a specific pH gradient basically. You can say 2, 3, 5, 7, 11, 12. So there is a pH gradient basically. So that will help the protein to as per the pH. As soon as the specific protein reaches its pI value, it will have no charge. So once it is no charge, it will be unable to move ahead. So protein reaches isoelectric point pi, and as soon as it reaches, so what is pi? It is the pi of the protein does not move an electric field owing to its lack of charge. Uh, how exactly pi works? But so as you all know, proteins are made up of amino acids, but each amino acid will undergo this specific reaction of positive and negative charges depending upon their pH or depending upon the OH minus ions available in the solution. So depending upon that, the at a lower pH, this is at a lower pH. If you can see at 2.34, so at lower pH, what is the case? There are more number of H plus ions, right? In an acidic pH. So because of that H, all those H plus ions will come to the amino acid, and all H will be filled. And because of that NH3, there will be a positive charge. That generally in the acidic condition, proteins are positively charged, and there will be a point at which H plus ions and OH minus ions at a state where this specific protein, one of the H plus ions will be lost because OH minus are increasing, right? As we go towards the basic pH, so OH minus ions are increasing. So one of the ion gets removed, and similarly there are similar number of H plus ions as well, and because of that there will be NH3 plus as well as CO minus. Well, although one H ion is removed, so this is having positive charge as well as negative charge, indicating both are minusing each other, and there is no net charge as such, and net charge becomes zero because one is plus and one is minus. Plus one plus minus is equal to zero, right? Like plus 1 minus 1 is equal to 0. So that's how this specific amino acid gets a neutral charge. Similarly, as soon as the pH increases at that specific pH, the other H plus ion also gets removed because it wants to make OH minus ions, and because of that there is no H plus ion here as well, and there is only minus ion which remains in COO minus, and because of that the amino acid is negatively charged. So here we are just taking an example of one specific amino acid, but as proteins are made up of n number of amino acids, so accordingly each amino acid will have a positive, negative charge depending upon the pH, and overall there will be a point at which whatever proteins that whatever amino acids that protein is having, it will fall up into like similar. For example, if it is having 200 amino acids, 100 will have positive charge, 100 will have negative charge, and they, whatever is that value at which it is having similar number of positive ions and similar number of negative ions, then it has a neutral charge, and that's the reason that protein will be stopped at that specific location, and that's how the separation happens. So it depends upon each amino acid basically, and each amino acid, then accordingly the overall charge will change for a protein. I hope you all are getting it. So this is how the separation is here. Maybe then it is net positive charge. So as soon as it moves ahead, there will be no net charge and it will stop. So this is it. They like it is in a similar. Basically, it is depicted in a different way. So here initially it is a positively charged protein. Then as soon as it moves ahead, here is the, so for example for the green one, sorry, for the red one, so as it is moving, it is having positive charge, this protein had a positive negative charge at this specific location, so it stopped. Similarly, for the blue one, it had a positive negative charge at this specific pH, it stopped. And as soon as it goes ahead, and we'll get a point at which this specific protein is also becoming neutrally charged, and that's how this specific protein also stopped. So that's how the separation happens in terms of isoelectric focusing. Explain how isoelectric focusing is performed in general. But what are the steps we will have to follow in order to perform this experiment? That is the first dimension of 2D gel electrophoresis. So first step is rehydration. So what happens in this specific step is proteins extracted from the previous step. So initially you will be doing the lysis, which we discussed in week 2 basically. So once you have your protein sample, may it be a complex mixture, you don't know what all amount of proteins are present, but you know that you have extracted protein. So in your sample there are proteins, you don't know what all proteins might be present. Is it a mixture? How many number of proteins are present? You don't know, but you have a sample. So once you have that extracted sample, you will rehydrate the IPG strips, basically those strips which are these strips basically, these strips before loading the protein. So you'll rehydrate those IPG strips overnight in a reswelling tray at room temperature itself. So once you rehydrate them, so it can be done. It is done in two ways: passive rehydration as well as active rehydration. Passive indicating there will be no voltage applied, only the uh, rehydration buffer is added. So it's called as rehydration buffer or IPG buffer. So if you are not applying voltage, it's called as passive rehydration. And if you are applying voltage, it is called as active rehydration. Then once the rehydration is done, you will, after overnight rehydration, you will add the mineral oil over to it. So you will basically overlay mineral oil on the rehydrated strip. So that's what rehydration step is. So basically you are rehydrating, making it active basically. Initially the strip is not active. If you are not rehydrating, it is not active for performing the experiment. The proteins will not get separated, or the charges on the strip are not active. The pH values basically. So once this method is followed, it becomes activated. So this is what the initial part is. So you need to ships are supplied. What all different points you can remember out of these is, so IPT strips are supplied dry and stored at temperature -20, which is generally the storage location for IPT strips. And samples should be loaded on the strip after thawing. Like as it is kept at -20, you need to bring it out and uh, thaw it for some time, and then only samples should be loaded. And samples can be loaded either in cup loading, direct loading. So there are different ways of loading as well. And ships should be covered with mineral oil to prevent evaporation. So those strips, there are different ranges of strips available. So depending upon your protein of interest, you can accordingly use those specific pH strips, like IPG strips basically. So if you know that your protein is the sample you are interested in, that specific protein has a PI of around 6 or has a PI around 5, then accordingly you can use this range pH itself. If you know that if the PI of your protein of interest is above 7, then you cannot use this specific strip. So you'll have to use this specific strip so that your protein of interest is present in the initial first dimension, or else in the first dimension itself it will go out of the range itself, and proteins will not be there in the IPG strip. And there are different lengths available, smaller sizes as well as bigger sizes, 7 centimeter to 24 centimeters, and each are capable of accommodating. So there are different IEF units as well, wherein these strips can be accommodated, and larger gels are recommended. Why? Because generally you will be doing two-dimensional, right? So generally people prefer 24 centimeter one, because larger the gels, the resolving becomes much more proper. Because if this much is the space and if there are this much number of proteins, it will move and it will be nearby together. And if this is the 24 centimeter we are available, then each and every protein will be separated enough that you can visualize it at least. Or else it will be very difficult to understand whether they are separated or they are not separated. So, in terms of loading, what kind of staining amount you need to use depending upon the type of strip you are having? Per 7 centimeter, you need to use around 160 microliter of this specific staining dye. Or there are different staining dye. We'll be talking about this, but this is just to say that what volume you will be needing according to the IPD strip you use. So that's what first dimension separation is all about, wherein separation of is in IF is on the basis of isoelectric point, and sample loaded strips after overnight rehydration are focused on isoelectric focusing by passing current, and protein mixes migrate in electric field and come to rest once pH is equal to Pi. So that was the first step. Then we have done the rehydration stuff. We have run the proteins as well. Once the proteins are separated, then that specific strip needs to be equilibrated. So, how do we do the equilibration? Because we need to do equilibration before going towards the second dimension of 2D gel electrophoresis. So, in the first dimension, after once the proteins are run in the IPT strip, you need to equilibrate. So, how can we equilibrate? Let's discuss. So, after the first dimension, a conditioning step is applied, which is called as equilibration. So this is applied to proteins separated by IEF, sorry, and the coats protein. So what happens with this? So in this specific step, you are coating the protein with SDS so that protein will gain negative charge. And once they get negative charge, they can go towards the positive charge in SDS phase, and the separation can now become on the basis of molecular weight. And if we are not charging it, we'll not be able to separate it. So that's the reason we add SDS first. And that SDS, what it will do, it will also cleave inter and intra-chain disulfide bonds, and it will also be responsible for alkylate sulfidyl groups. So, in terms of equilibration, there are different steps involved in equilibration. The first step in equilibration is you will be saturating the strip with SDS and reducing agent. So, remember, first you are doing uh, reducing. First step is about reducing the IPG strips. So, in that, reducing agents involve urea, SDS, DTT, etc. So these SDS, DTT, and urea, these are the three major ones which are responsible or which act as a reducing agent, whereas all these this HCl and glycerol is trying to maintain the buffer as well as the density of it. Then the second step is alkylating. So alkylates residues by using DTT to minimize vertical streaking. So when you do 2D gel electrophoresis, you will run the SDS vertically, wherein you need to have a separated bands, right? Once you are separating them. So if you want them to be separated properly, you need to alkylate them with DTT, and basically that will help in minimizing the vertical streaking. And if you are unable, if you are not doing this, then it will basically you will have a streak of the proteins getting separated. You'll not get the separate bands, and you'll unable to determine what is the protein and what is the molecular weight, and that it will be a fluff experiment basically. Then iodoacetamide is used in order to prevent protein reoxidation, and these specific concentrations of reducing agents or second in secondary equilibration also these are used, and equilibration time is twice per 10 minutes each. So what happens in this specific reactions, like in this first equilibration and second equilibration? So in first equilibration, what is happening is whatever are the disulfide bonds, those disulfide bonds are broken, and that's how the protein gets separated in the first step. And in the second step, what happens is those specific proteins will have a specific um group attached to them, that is this iodoacetamide. This basically will prevent the protein from reoxidation. So if it is reduced, if it is separated, so once we add this specific group, it will not join again, right? So it is preventing them to reoxidize, from preventing them to not to reoxidize. So that's the reason we add iodoacetamide, which adds it to the sulfur group, as we have broken the disulfide bond here. So once it is added, so that's how the prevention is being done from oxidation. So that's the equilibration step, and that was something which was discussed in the part one of the search lecture. Then from part two, or the second lecture of week three, sir discussed about the second dimension. So what have we have done till now? So we have performed the first dimension. Once first dimension is done, we need to equilibrate that specific strip, and we discussed about the equilibration. Once the strip is equilibrated, we can proceed ahead for the second dimension. Uh, so for doing second dimension, the sir just gave different what do we say, animation kind of a different animations of performing this specific experiment, as well as some practical demonstrations. You can go, if you have all have not went through those videos, at least you can go through how exactly practically it looks. But still have tried to cover crop certain images and show you, so that it becomes easier. So this is how the gels are made, and this is the gel, if you can see here. So once that is there, so this is the equilibration step, you can say. So that specific strips are soaked in the buffer. So whatever buffers we were talking about in first equilibration, second equilibration, that's how we do. And after keeping it in the specific racks, there will be SDS gel in below this. So you'll be keeping that specific IPG strip, and you'll also need to load the ladder here. If you can see uh, a dark blue stain here, that's nothing but the ladder, which will help you to determine the molecular weight. Then you can keep it in the tank buffer. This is how the tank buffer looks. So this is practical demonstration which Sir tried to show. So what does those first equilibration step and second equilibration step do? We already discussed its chemistry, but we can discuss a bit more regarding it. So what does SDS do is it is basically giving negative charge to the protein, so that the proteins will be able to separate from negative charge to positive charge in terms of electrophoresis unit. And this separation is on the basis of molecular weight. Higher molecular weight will be coming resolving a little slower, whereas lower molecular weight proteins will move faster and resolve faster, basically. So that's what it is. So it is a powerful technique for simultaneous separation of thousands of proteins. In second dimension, what we are doing is we are separating proteins depending upon molecular weight, and no stacking gels are required in second dimension in terms of 2D gel electrophoresis. And if you are doing only SDS page, if you are doing only SDS page, then it is required. But if you are doing uh, after using IPG strips as a second dimension in terms of 2D gel electrophoresis, then stacking gel is not required. So once that is added, once the strip is added, you visualize the gel. For visualizing the gel, you need to stain it first. And once the stain is done, so staining is generally done overnight or around 18 hours, so it stains immensely. And you will also have to de-stain it, so that only the proteins are getting stained, and whatever is the gel part of it, and there are no proteins in those parts, those specific parts will be de-stained by using de-staining method. So how exactly you'll be doing staining and de-staining? So in terms of staining, there are different stains available. One of the commonly used stain is commassive blue. So most commonly used stain for protein detection in terms of SDS page is commassive stain. which is also considered as a biosafe comassive stain, and it is ready to use. It is a single reagent protein stain where it stains something like this. It looks blue in color, and it can be disposed as non-hazardous waste as well. So once your gel is run, basically you will be adding that into the comassive blue solution. Once it is stained, you need to wash them by using a destaining solution. So destaining, we'll talk about that later. So let's talk about different stains which are available. One is comassive blue stain. Other is silver stain. So this silver stain is basically more sensitive than comassive blue stain. But for this specific staining method, you need to first fix the proteins first. Like as soon as you have run the gel, you cannot directly stain it. So once SDS page is run, you cannot directly stain it if you are doing it with silver staining. You need to fix the proteins. For fixing proteins, you need to add basically methanol and acetic acid and incubate the SDS page gel in methanol and acetic acid. So that activates the proteins in order to get stained from silver staining. So what is silver staining? It basically has silver amine complex bound to tungstosilic acid. So that's how the solution is about. And here the Ag ions transfer from this specific acid to proteins, and that's how the staining happens. And there is also formaldehyde in the alkaline solution, and it reduces silver ions to metallic silver. And that's how it stains the protein. And if you want to, once the staining is done, and you need to stop the specific reaction, and that reaction is stopped with acetic acid. So this is how the gel, you need to activate it first, like using methanol and acetic acid. After fixing, you'll just add the silver stain onto it, above it, and this is how the reaction is happening. And if you want to stop the reaction, you need to add acetic acid. That's how silver staining is. There is cypro-ruby stain as well, where this is a fluorescent stain, and it is easily visualized with simple UV or blue light. So basically, we'll just need a trans illuminator, and after staining, we'll directly visualize on a trans illuminator. Just a minute. Yeah, you can directly visualize on the trans illuminator when you use cybo green as a staining stain for your stain for staining proteins. And this specific stain is also has a capability to detect glycoproteins and even lower molecular weight proteins as well. So that is good if your protein is very much uh, of a lower molecular weight. You can use this specific stain. So this is how the fixing is. So once your gel is uh, uh, run, like the SDS page gel, you need to fix the specific protein. If you are using specific dye, if you are using comassive blue, then this specific step can be skipped. So how do we fix it? We're using 50% methanol and 7% acetic acid. Once it is fixed, you will add the specific stain, this specific fluorescent stain for incubating. Then after that, you will after you just need to remove the stain and wash the specific amount of stain which is not needed. Only proteins are something which you want it to be stained. And after washing, you can visualize in the gel scanner or transilluminator. For this specific stain, there is some excitation wavelength and emission wavelength, and you need to set those specific parameters. Once you set those specific parameters, you can visualize the gel. So these small dots are nothing but the proteins. There is one specific staining technique which is called as ProQ diamond dye, which is a fluorescent dye again. So there are two fluorescent dye we are discussing. One is Ruby one and one is ProQ. So this specific stain can also detect phosphorylation, the post-translation modification which we are talking about, which we talked about in the earlier slides. So if any of the protein is having some post-translation modification, if you want to check them, you can use this specific staining technique, and this will enable to help you detect those specific proteins, and it is suitable for use with electrophoretic techniques, and it offers sensitivity in nanogram levels as well. So that's how good this specific staining technique is. Similarly, you need to fix it for this staining technique, then you will stain it, then you will wash it, and then visualize it at certain excitation and emission wavelength, and this is how you will get the image of the ProQ diamond stained gel. Then people also use dual staining technique. So dual staining in the sense they'll be using two specific dyes for staining. So it is ProQ diamond or other staining detection stains can be combined with other staining procedures such as Cypro Ruby. So that's how you will detect it. Then dual staining allows you for more than one detection protocol on a single gel basically. So you will be having single gel itself, and in that single gel, you will be staining with two different dyes, like whatever the dyes we discussed. So that's nothing but dual staining, and it basically allows more than one detection protocol on a single gel. So this is just uh, um a overview of all the staining technique: the Comasive blue, Biosive, Comasive silver stain plus Ruby, as well as ProQ Diamond, etc. There are a number of it, and their sensitivity ranges. So these are more sensitive. If your protein is very low concentrated, you can use these techniques, and if it is concentrated, high concentrated, like good enough concentration, you can use this commonly used stains as well. And so these stains are MS compatible as well. So if you are further uh, approach is doing mass spec, then you can use this staining technique and give it for mass spec analysis also. So that's an overview of about staining. So that's what we discussed in the second lecture, and these are few references which will help you to find these different in detail about different staining techniques or in general about 2D gel electrophoresis. So you can follow this any of this book. All of this will be having this thing. And after that, in the third part or the third lecture, so I talked about different scannings, gel scanning, different analysis methods. So we did talk about destaining as well, right? So this washing is nothing but destaining step, okay? So don't get confused between destaining or washing. So the word destaining may basically you are washing the gel. So this washing is equivalent to that of destaining. And once that is done, then you will visualize in the gel scanner or a transilluminator, whichever is applicable. Are you all getting it? Yes. Till now, everything is fine. Yes. Okay, thank you. We will do one more part of the lecture and then we'll take a break of 5-10 minutes or so. Yeah. Then, sir discussed about analysis. So, so once the gel is stained, you will analyze it. So for analyzing, you need to visualize the gel. So in order to visualize, you can use transilluminator, you can use chemidoc. There are different instruments available just to visualize the gel and visual interpretation if you want to do and compare the results. You can do all of those stuff. Then densitometry or the typhoon variable mode imager. So these are two maybe you can say a little advanced instruments in order to perform analysis. Like if you don't want to go till mass spec, so mass spec is much more accurate though. So if you don't want to go till there, you can still do a good amount of analysis using these kind of instruments. So, so densitometry. So basically, this specific instrument will help you to find which specific protein is over upregulated or overly expressed, because it basically determines the density of. So once, if you have seen any gel image as such, so each specific spot or each specific band, it will try and determine how much amount of protein it will be there. So it will be giving relative readings basically. If it is much more denser, then it will consider as 100%, and the smaller proteins, it will accordingly relate with that specific dense protein, and accordingly tell you that this is the amount of protein is highly dense, or this is something which is having lower dense compared to other proteins. So that's how it will try and determine this molecular images, try and determine stuff. So this is maybe as an example how software looks, as one of the examples. So 2D gels are scanned using a scanner, and images are analyzed using various software. So that's what I was talking. This is just an example as one of the software. So what does software generally help you in? Is it helps you in spot identification. So if these are any specific protein, it will try and help you to identify those spots. It will try and help you to compare the gels as well. So you can run the same specific sample from one healthy individual and from diseased individual, and at the same time and following same kind of routine, and you can just run their samples, and basically a healthy control and a disease control will have some proteins here and there. Then you can basically find what are those proteins and what that specific disease is leading to, what changes in the protein level. So that's how what you all can determine. That's the application part basically. Then how do we determine that? By overlaying of those images, and also you can zoom those specific uh, like if this big is the gel, you can just zoom into your interested proteins. You can crop those, you can overlap them, you can do statistical analysis as well, which one is highly expressed, which one is lowly expressed, and all of those stuff. So these are certain software application, like it has a crop tool. You can take the desired image, whichever you are interested in. You can zoom into one of the specific area as well for further analysis. So here, although you're looking at it in a very separated manner, and you will be like, why there is a need of zooming or why there is a need of cropping and stuff, because it is clearly visible. That's what you people might be thinking, but that's not the case in the real experiment. The proteins are very much close together, and because that's how dense our protein pool is, or our protein concentration is, and that's separation becomes. So that's the reason we were talking initially that we need to have a higher, larger gels and not 7 centimeter. It's better to have a 24 centimeter, because that's how separation will be easier. And so once we zoom into that very much specifically, so we would be able to see this much amount of separation. But if it is unzoomed, it will be like very much close together, and it will be difficult to analyze. So that all those can be made easier by using software. So it can help you in image overlaying, and that will help you to understand this in this gel it is overexpressed, in this gel it is lower expressed, and in this gel this protein is present, but in this it is not at all present. Because in disease state there might be few proteins which is not able to express, or which is some there is some protein which is not there in the healthy individual. All those can be determined using these techniques. So there is spot analysis as well, like each specific protein. It is possible to obtain a physical and statistical parameter of each spot on the gel itself, and it allows to compare between spot by spot basis as well. That's spot. So this is how complex a gel looks basically. So this is the controlled one, healthy control, and this is the diseased sample. So you can accordingly see, like if this is comparatively higher compared to this, then there are some few proteins here which are not present in the diseased one, right? So there are few proteins, like this is one of which we can clearly see there is something in the control one, but there is this specific proteins are not present here. So that's how you can compare accordingly. So software, these are few softwares and these are few websites which sir has given. So these are different softwares which are available. You can use any of them to try and so if you are having any specific two-dimensional gel, then accordingly you can analyze them using this software. So this is how it looks. So once you do the matchmaking or gel matching of two, like how they will give you a certain score basically to check how accurately it is matching or uh, how it is not exactly matching or it is matching basically that's what how accurately it is matching. And for every spot it will try and give one specific ID and the intensity of that specific. So for example, this will have higher intensity because it is larger in size as compared to this, and it will give this will have lower intensity. So accordingly to each and every spot it will give an ID, it will give its intensity, it will give you an area value, its volume, so percent volume comparing with the others, and it some of its features as well. So that's how uh, the software will help you to determine spot by spot. You can also perform certain statistical analysis using that in the software itself. And like for here in spot 5, here Spotify similarly. So after so this is the first dimension running from, although it is blur, I'm just explaining from pH 3 to pH 7 they have used. So proteins are run here initially, then after that it is placed on SGS page and your ladder is added and it is separated, right? In the vertical direction. So here you can see in the gel one, the PI was 4.8 here, then the PI was 4.9 because if you can see it is slightly towards right hand side, the yellow one. So they are trying to compare these two spots basically. And accordingly, this is around 40, 43 uh, molecular weight, whereas this is slightly lower than 42. So it is trying to compare them. So that all those analysis can be done using software. So that's how it will help you to study global protein expression. It will help you to study differential protein expression. It will help you for resolving proteins from complex mixture as well as isoform. All those can be done. So this is just an overview of each and everything. Just you will be having two proteins, for example, in one condition, in other condition. You'll just use the first dimension by doing isoelectric focusing will separate the proteins. Once those are separated, you will be doing equilibration step, which is reduction and alkylation. Once equilibration step is done, you'll place it on the SDS page gel. You will run them. After running them, you'll have to do any kind of staining here. Once the staining is done, you need to visualize using um, those different softwares and analyze using different softwares. So once you are able to analyze, you can pick the protein of interest, and that specific protein of interest can be given for mass spectrometry for identification of that specific spot as well. So it's the same thing. You can do spot analysis. Whatever is the protein, extracted protein, you will do run the SDS page, two-dimensional gel electrophoresis, determine those uh, proteins by using different analysis software, and that's so this is it's basically a cycle. So it helps you in separation, identification, quantification of proteins, it helps in analysis of cell differentiation, it helps in detection and identification of biomarker, it helps in protein purification. All these are certain applications of 2D gel electrophoresis. Then sir went ahead to talk about different applications of it. One of the case study he talked about malarial infection, and he discussed about that. Maybe we can deal with case studies after taking a break of around maybe 5 to 10 minutes. We can, so now it's 3:55 and we can start at 4:05 or something after 10 minutes. Fine. We can take a break of 10 minutes. Then we can talk about case studies and then we can discuss few questions. If you are having certain questions, we can discuss on that. I am having certain previous year assignment questions. We can just discuss on that too. Okay. So now it's 3:55. We can start at 4:05. Thank you.
ब्रेक नहीं बोलने का अरे मैसेज नहीं बोलने का ऐसा टिकट रखा बोला
हाँ हाँ हाँ, देखा मैं।
क्या दुकान में हाँ बस होती है गाने बोले गाना बोल नहीं तो सब खेले गाने में बोल गाना बोल 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 वो सही नहीं है लेकिन हम बोल निकाल आप बोल ना एक बोलते एक बार बोल देती डांस में गाने करते गाने अभी वही गाने में डांस से करते तो वे कैसे वही गाना बोलते हैं बोल देती ये देखा नहीं अरे क्या देखा सेंड करो मुझे व्हाट्सएप पे भेजो ए बाबा बोलने के लिए बड़े बोले लेकर आ तो चौधरी आने का कि नहीं ये वो वाला वो भूत वाला क्या था उधर भूत वाला किधर है भूत वाला किधर है भूत वाला भी था कौवा कौवा भूत वाला क्या था वही भी एक था वो था ना तेरा वो दो नहीं अरे ऊपर बैठ गया था कौवा कौवा करके कुछ था ना
10 मिनट का ब्रेक दिया हम।
ये जोश जोश में आ गया तेरे से बात करने के लिए
हाँ, ये आ गए। Superman, Superman, right? Superman. Woo!
सब लोग जो मिटा है वही रख देने का और क्या
She's sleeping. हेलो, नहीं चालू है, ब्रेक दिया हूँ। लेक्चर नहीं, ब्रेक दिया है। सो जा ना तू थोड़ा बाल हो गया आग तेरा अपन सो जाके आते हैं। Oh, fuck.
अरे तो पैसे देना पड़ेगा तुझे फिर राखी बांधा तो मुझे भी बांध
Hello. I think we can start. I hope you all are also back. Yes. So in terms of applications, so in order to explain you all applications, I discussed about two different case studies. Did I on the mic? So yes, as well as what? Yeah. Yeah. So Sir talked about gave overview of two different case studies. Basically, one case study was about malaria. So how can we determine whether if a person is suffering from malaria using proteomics based study basically? And so you need to have an healthy control and you need to have a person who is suffering from malaria. And if malaria also like which kind of uh, parasite is exactly affecting, all those can be determined using proteomic analysis. So you'll need serum of that specific, if you all know the malaria life cycle. So in the serum, you will get those specific proteins from which are related to malaria, which happens because of Plasmodium vivax. So Uh, so you basically need serum and you will do the serum proteome analysis and basically it will help you to identify the disease pathogenesis and host immune response as well. So you'll need a healthy control, you'll need a Vivax malarial sample. So these are two different individuals. One individual is healthy, one individual is suffering from malaria. So you will do all those analysis, then you'll get the gel, right? Like you will take this sample, run IPG, isoelectric focusing, then you will run the SGS page. Once that is done, you will stain the gel. After staining, you'll get the uh, uh, gel basically of separated protein. Then you can take this one specific band or one specific protein as well for determining, for doing data analysis, for doing MS analysis, etc., and identify which protein, and accordingly that specific protein can be validated and characterized as well. So this is a short overview of each and everything. We just discussed this initial part of it, like in much more each and every specific detail about it, as well as a bit of data analysis part. As we said, like if you want to determine much more about identify and characterize the protein also, here you are just quantifying कि the protein is there and this is the relative amount it is there. You don't know exactly how much. If that you just know that that protein has certain molecular weight and this specific protein is present. You just know that, but still you cannot exactly say that that protein is the same protein which I am thinking or which I am supposing. So you cannot say that by doing this data analysis. You can talk about that only when you do mass spec analysis and then you do the validation and characterization of that specific protein. Then only you will be able to say that whatever band I can see here of that specific molecular weight is the same protein which I was thinking. Here you can just say yes, it might be because it is having same molecular weight and it is coming from the sample of interest. So if all those things are matching, then you can just say that that might be the possibility, but you can still not accurately say that that's the for sure protein. That can only be said by doing mass spec analysis, and after that also you need to validate it. Then only you can say that okay, that's the protein which are looking on the gel is the same which I am proposing or saying. So there are certain serum sample processing initially. Basically, you need to remove the abundant proteins. Like if this whole thing is a serum protein, so all these small things are serum protein. Out of these, there are certain serum proteins which are very abundant, like IgG, IgGA, albumin, all these are you can see, so this specific circle indicating the abundance of that. So basically you need to remove the abundant proteins because you are not interested in them, right? You are interested in those Plasmodium vivax related proteins. So you can remove that. So removing can be done in a different methods. So once you have the serum sample, you can do analysis of the whole crude sample. You can do desalting, you can do sonication and desalting. You can remove the abundant protein by desalting itself. So all these techniques can be done. So how can you extract proteins from the serum sample? So you are having serum sample uh, which you need to precipitate, and 4 volume of cold acetone is added initially. So this is so in week 2 lecture we did discuss about protein extraction using TCA as well for protein precipitation. So if you all remember that, or if not, then you all can go through those week 2 lectures where we talked about how exactly protein extraction is done. This is just to revise once again. You can incubate at -20, then centrifuge at 15,000 G, then 1 ml of cold acetone is added again in order to precipitate the protein. And or you can, after that, it is incubated for 15 minutes on ice itself. So everything should be done on ice because proteins are not that stable in room temperature. So in colder temperature, proteins are bit more stable. So generally, you need to do all protein extraction procedure in cold conditions. Then after that, you can centrifuge it and acetone containing supernatant is removed. So in terms of number of spots found by using different techniques, so this is a crude serum which wherein the it was not extracted after desalting. A bit of amount of proteins got increased after desalting and sonication. More amount of spots were observed, and similarly desalted, sonicated as well as depleted. So if you do all of those techniques, the number of proteins increased. So basically, this graph is actually, this image is actually telling you that if you the effect of different sample processing, indicating different extraction techniques. You can just, if you are loading this directly or using only one step, using this both the steps or using all the steps. So if you process it, the pre-processing is accurate enough, then you will get more number of proteins or more number of spots will be visualized on the gel. That's what this specific graph is talking about. So you need to optimize depending upon your protein of interest. You can directly, this can be your control basically, adding the whole serum sample. Then you can use acetone precipitation, you can use desalting, you can use sonication, you can use triazole extraction method. So there are n number of different methods. Few of it we discussed in week 2 lectures. You can look into that. So according to the type of extraction. Basically, this is also talking about the effect of processing. So in acetone, this is how there is some spreading stuff. If you use just acetone, if you use TCA acetone, a bit of separation was achieved, but still it is not that accurate. Although the number of spots observed in TCA acetone is greater as compared to that of just acetone. Similarly, using crude sample or depleted serum sample. So if you pre-process it by depleting, the separation becomes much more accurate. So here it was not at all separated by using crude serum sample, but because of that pre-processing initially, uh, it got separated here. So that's how important pre-processing is, the protein extraction part which we discussed in week 2. Similarly, in this lecture we talked about staining technique. There are few staining techniques which are less sensitive, few which are more sensitive. So using this technique around 919, whereas here with 995 spots were determined. So silver staining also detecting few proteins which are lower concentration, even 1 nanogram level of protein are also determined using silver staining technique. So depending upon this, so number of spots or type of it achieved. So sonication, the sonic in all of them, the amount of protein loaded is similar in these two, but in this one, the amount of protein loaded is less. That's the reason you can see a little not so dense band compared to this, because the protein concentration here added is higher. The strip size of all of them is same. All of them are stained with chromacy blue. but the number of spots identified are different in all of them. Why this is different? Because of the technique, because of the crude sample, because of sonication, because of the TC acetone or trizol method. So because of the initial technique used, the number of spots observed by the software and by refinement is this. So these are changing because of the initial pre-processing. So that's how the way you are making samples. That's the reason the whole week one, week two lecture were on sample preparation itself, because that's how important sample preparation is. That will completely change your final outcome of your results. So here comparative analysis were done. So these many viral malarial samples, these many healthy control samples were taken, and the healthy control had such kind of suppression, and whereas the disease one had such kind of suppression. So although visually we are not able to determine anything drastically here, but after doing software analysis, differential express spot analysis from each and every spot, so it will look into each and every minute detail. Like this will be able to zoom into it and try and check what are the differences observed here and here. So all those minute analysis can be done in the software, and depending upon the amount of staining it has taken, it talks about the upregulation and downregulation. So there might be some spots which are denser and highly like darkly spotted, whereas few will be smaller in size and not that densely spotted. Then we indicate at the same location basically. At the so this location itself, this location itself. So if you see clearly here, this is maybe a bit more densely there compared to that of this. So this in the disease control, it is getting upregulated, and here it is at a normal level, we can say whatever. So this all those, so here visually we might not be able to say it that accurately, but with software it helps us to do that, and it will help you to find which proteins are getting upregulated, which are getting downregulated. Similarly, that's how the proteins, few of them are having fold change depending upon the type of protein. So altered protein expression levels in Vivax malarial patients, there were few which were upregulated, there are few which were downregulated. Then they later did mass spec analysis as well, wherein they tried to determine what kind of protein it might be. For example, they have said this protein is getting highly upregulated. So they just choose this one specific protein and tried to do their mass spec analysis and tried to check which protein is that. So then they identified that these were the few proteins which were downregulated, and these were the few proteins which were upregulated in the malarial patient as compared to that of a control patient. I hope you're getting this. So that's how the application is. Then this indicates that if you're doing just blood test and if you see these specific proteins are down regulated than the normal level, then you can say that that person might be suffering from malaria. Similarly, you find this protein in the blood test and so found that that is getting upregulated, that might indicate that. So that's how it will. So these studies help you to find biomarkers as well. So these are few references. Then, and the final lecture, fifth lecture, sir gave few more case study examples, like effect of drug treatment on malarial parasite, which we talked about. No, not drug treatment we talked about, and the plasma proteome analysis of SARS disease, like COVID. And sir also talked about certain challenges with two-dimensional electrophoresis. So here one more case study where definition of coatom action in Plasmodium falciparum. So this is a drug basically. So what is a coatom? It is a combination of artemisinin derivative and artemether with lufepantrin. So these are two specific drugs which are given for this specific treatment of Plasmodium falciparum malaria. So drug of choice for all cases of non-severe malarial worldwide. This is the drug which is generally given. And how does it act? So its drug action is mediated specifically through its endoperoxide moiety. So they initially tried to check the Uh, so this is how the falciparum looks. Then that's the late ring stage in the That's how the life cycle is. You all might be knowing the malarial life cycle. And you'll culture the cells and you'll try. So here they try to find the growth phase of that specific Plasmodium falciparum. Then they took that proteome of this specific alteration in Plasmodium falciparum. So they, while growing this, they have given these kind of drugs, and then they try to check the effect of those drugs. So that's what the study is: effect of drug treatment on malarial parasite. So this is the malarial parasite, Plasmodium falciparum. They have given these specific drugs and try to check their effect in their proteome level. So initially there is fractionation of this, like from Plasmodium falciparum, we are extracting proteins. That is parasites are isolated from host erythrocytes, as the parasite generally infect the RBCs, the erythrocytes of the host cell. Then host cells RBCs were taken, and from that parasites were isolated, and then they were washed, solubilized, and then the fractionation of that was done, like the lysis of using lysis buffer, and the protein was extracted from Plasmodium falciparum. Then the initial isoelectric focusing, then after isoelectric focusing, gel electrophoresis was done, and this is how the separation was obtained. So once that is obtained, people, so drug specific alteration to falciparum proteome was checked. So this is the two drug which is having two of these drugs. So if we give that at the ring stage, so which is inside the erythrocytes, right? If you just recall the malarial cycle, so it is a reaction with late ring stage of Plasmodium falciparum. At this specific stage, this specific drug infects and glycolytic enzymes are released. So you will be able to see certain glycolytic enzymes in the 2D gel electrophoresis pattern. So altered protein expressions were observed following drug exposure. So with this drug exposure, these were proteins which were upregulated, and these were certain proteins which were downregulated with this drug treatment, whereas it was upregulated with this drug treatment. So altered protein expression level following drug exposure. So stress response proteins were upregulated and glycolytic enzymes were upregulated, as well as were downregulated using this specific protein, but with this specific protein it was upregulated. Similarly, this specific drug and this specific drug, stress related proteins were upregulated. I hope you are understanding the image. So basically, they tried to study the effect of each and every drug and different Plasmodium falciparum associated proteins, and they found that the investigation of alteration to the parasite proteome was induced by two components of this those two specific drugs, and they tried to establish a relationship between pharmacologically relevant concentration and time of exposure for these two specific proteins, and that's how they found which proteins are getting upregulated and which are getting downregulated, and accordingly, if a person is getting cured out of these two specific drugs, what exactly is happening to Plasmodium falciparum and how why it is dying. So they are, that's how these experiment will help you to understand. So that was one another case study sir talked about. Then SARS was the other case study, which is trying to check the plasma proteome analysis of SARS study, where 2DE as well as mass spec was used. So as you all know what SARS is, it's a respiratory disorder. It has certain glycoproteins and stuff which actually uh, do all those symptoms like COVID, which we all know like very much in detail, as it was whole 2 years was COVID was that specific phase. So I'm not going in detail about this. So there is spike proteins. You might be knowing that. That's what binds with the specific receptor inside the lungs, and it helps. It causes respiratory disorder. So they took four SARS patient and six healthy patients. Then all those specifically were run, and the isoelectric focusing as well as 2D electrophoresis. Once that was done, they did trypsin digestion forward, so that ahead, so that proteins are digested properly. So this is how the proteome analysis was. observed. So you can clearly see that in healthy control, this is one specific protein which is highly expressed, but in diseased one it is low, it is not that expressed. Similarly, this specific protein is overexpressed in the diseased patient. Similarly, this specific protein is overexpressed here. So you can here for in terms of this image, you can do it visual analysis as well. But the software will give you exactly how much it is higher than this and how much it is higher than this. Accordingly, we can know by software analysis. Then ahead, they did mass spec analysis, where they tried to find what are these specific proteins. So you can just cut this part of the gel and isolate protein out of it and do mass spec analysis. That's how it is done. And they also tried to check one of the protein like PRB2, redoxin protein something. And this protein, so normal individual as well as patients. So they found that it is found in two forms, 22 kilodalton as well as the disulfide together 42 kilodalton. And they both together will so in the reduced condition, the disulfide as well as the monomer form of it were observed in one of the patient, whereas in non-reduced form, only one was found and there is no band at 22 kilodalton below here. So that's how they found, like Western blot analysis will help you to understand more about this one specific protein in the patient samples. So what happens is basically because these acute phase proteins are in excess when a person is suffering from SARS-CoV or SARS related diseases. So so when we are studying this kind of diseases, we are interested in these proteins itself, like acute phase proteins. Then those was three different case studies I talked about and tried to make you all understand where all this specific technique can be applied. And similarly, what all certain challenges which we observe in terms of two-dimensional electrophoresis is. Uh, so these are certain examples. So this is a very good example. Like these are very separated proteins are achieved, and each and every spot is kind of visible, and these spots can be visualized from the software. So that's how. So this is one good example. Similarly, this is a good example of one of the bacterial B. subtilis proteome. Like each and every spot is clearly visible here. So this is a good gel. But there are certain gel artifacts as well and gel to gel variations, sample to sample variation, the way you are doing electrophoresis, at what voltage, what current exactly. So all those will have some effect to the gel. Similarly, in terms of image analysis, defining spot boundaries. So you need to define spot boundaries while doing analysis. So if you are not doing that properly, your analysis might be different. Similarly, user bias, like some people will try and focus at one specific location and tell that. So user biasness can also be there in terms of data analysis. These are few challenges. So as there can be some problems which we generally face because of not pre-processing it or not equilibrating the gel properly. So all these problems might arise. One of the example like streaking problem, like this is how you will not get separated proteins, but all those proteins will be in just a streak. Similarly, some spreading can also be there, like if there is some trace impurity while doing in the buffer itself. So this is how the results can be there. If you use old TEMED while making the SDS page gel, this is how the protein can be observed. Similar, if you are using crude sample, it will have everything, and that's how spreaded gel you will be seeing. So these are certain problems which you need to tackle with while doing uh, 2D gel electrophoresis. So if you all follow properly those pre-processing steps, equilibration steps, similar what a specific voltage, and you are having control studies equally, then it will help you to determine or have a good study basically. If so, these are certain equilibration problems if you are not doing properly. Still, this is a good image though, but There is some spreading you can see here. The destaining might not be done properly. If you are trying to over focus, then also then some certain kind of gels can be seen. And there are different source of variations. It can be biological, it can be inherent, and or it can be induced as well. So it can be between individuals, between biological states. It can be uh, effects which can't be controlled and effects which can be controlled, like sample preparation, etc. Some biological problem and some technical. So these are certain sources by because of which you are getting certain variations like this. So these are can be possible sources depend because different samples they were and different they were belonging to different biological states. Then there might be some different proteome inside them. Technically, these are biological problems. Technically, maybe you are not able to control. There are some inherent problems itself. You are not controlled. There is something which just because you are not making the sample preparation properly, the pre-processing, the after processing, the spot boundary and stuff. So all those can affect your outcome. So the same thing, different sources, variation in sample preparation, IP district pH range, presence of salt, which is a contaminant, voltage applied, voltage you are doing, equilibration time as well. The each and every time also affects the gel, the quality of chemicals used, user biases, etc. So these are certain case studies and challenges I talked about in his last lecture. Those were the things. Uh, Screen is not visible. Is it not visible yet also? It is not visible. Is it visible now? Oh, you might have unmuted yourself and told me, right? Because I started lecture at 4:05 and your message is at 4:24 after 20 minutes. You might have unmuted yourself and told, right? Okay, just we'll just go through it faster. I already explained it. I hope you all listened to it, but in order to just to see them once, so I think we were somewhere here, case studies part. So I'll just rush through it once. So I was just talking about these sample preparations, these abundant proteins which we need to be get rid of in terms of serum, different techniques. This was the higher thing which was I was talking about. If you are pre-processing, if you add multiple steps and do good pre-processing, the number of proteins visualized will be higher. Then these certain differences I was talking about using the changing one pre-processing, we'll get a different kind of this separation which I was talking about while talking about crude serum and depleted serum. Staining technique, you can will visualize more amount of proteins with silver staining. See, you can see all these are of same samples, but change in the uh, pre-processing part, crude, sonicated, TCA. So you can clearly see the differences in the pattern of separation. Then we were talking about this, these samples I was talking about. This is a bit intense compared to this. So we can by using the software, you can talk about how what are getting upregulated, downregulated. You can choose one of them, whatever you want to study ahead, do mass spec analysis and check which is that protein. And they found that these were few downregulated, these were few which are upregulated. Certain references to different other case studies, cycle of plasmodium, Then they, after while doing the analysis, they found that there is a release of glycolytic enzymes. So they found certain enzymes which were upregulated, found which were downregulated by treating with certain drugs. So these two drugs, the combination of it, so some got upregulated stress response protein like glycolytic proteins. In this drug got downregulated, this drug got upregulated. Then about SARS-CoV, so here I was talking about healthy control and the diseased one. So you can see this is the protein which is downregulated here in the disease, whereas this is the protein which got upregulated. This is the protein which got upregulated. So if you want to determine which is that specific protein, you need to cut this part and send it for mass spec analysis. So some challenges we were talking about, some good examples of gel, some bad examples like streaking, and all this can be because of certain impurities, certain use because you used old timid while making the gel, some sample preparation issues. So some bad examples we talked about. Then what can be those sources for these variations? So that's what we were talking about. So I just went through it so that you all can visualize them as well. So I think we can go through the questions now. If, yeah, so let's go through the questions. So week three, so these are previous year questions itself. So in two-dimensional gel electrophoresis, now in terms of equilibration step, just recall equilibration step. So what do we do first? What do we do? We do reduction first or alkylation first? We do reduction and then alkylation, alkylation then reduction, alkylation and then reduction together, and none of the above. What do we do? Option A, there are so many, I have opened it. Maybe if I can just, so option A is right, reduction and then alkylation. Then which of the following components leads to clogging of immobilized pH strips and could form complex with proteins by electrostatic interaction? So this you all can still answer by just having certain logical reasoning. So which of the following component leads to clogging in immobilized pH strip? So that is a pH strip. We have added the sample. We are trying to resolve it, but what could form complexes with proteins by electrostatic interaction? What all things can form a complex with proteins? Yes. Should I show the question again? Nucleic acid can form complex. Yes. What else can also form? Do you think only one can form complex? I just make it this smaller. All of them, you guess? Yes, it's all of the above. Okay, so your protein sample has a lot of phosphorylated proteins. And in this slide, which of the following stain would you prefer to use? So we have studied the staining technique now, and the question says that protein sample has phosphorylated proteins. So what do you think? Which stain technique we should use? We just discussed about that. Yes, it is option A. You all are right. So now Sandhya is performing an experiment. perform protein extraction using trizol method, then obtained 720 micrograms of protein from the sample. So 720 microgram was extracted. Then she has to run 2DE using 18 centimeter strip and further wants to identify proteins using mass spec. Select the closest amount of protein she should be using to start her 2D gel electrophoresis experiment. So this we haven't discussed, but generally you need to have some like till uh, after micro milligrams of protein so that because while doing this experiment there can be certain losses as well. Then this answer is around certain 300 to 1 mg around, which is 1000 microgram. So I'm directly answering this question because we haven't discussed as such regarding how much amount of proteins are generally used. So as if you want to use it for mass spec also later, then a bit higher is generally recommended, which is around 300 to 1000. This much is not required as such, around 1500, but till 1000 it's fine. So if you're not doing mass spec ahead, then these concentration also work just to detect. But as you want to do mass spec analysis later ahead, then you'll need certain protein sample preparation as well of that specific protein. That's the reason you'll need a bit higher, which is around 300 to 1000 micrograms. Okay, which of the following pH range IPG strips would you use if your sample contains protein with acidic pI values? What do you think? I think this is a easier question. Which pH strip would you use if you know that it is having acidic pI values, your protein of interest? Answer B, 3 to 6. Yes. So Rohan, when visualized his 2D gelatophoresis gel after the gel staining, found vertical streaks in the gel. So it found vertical streaks. We just discussed what all can be the possible reason for vertical streaks in the gel. So which of the following factor may cause such streaking pattern in the gel? Low acrylamide, percentage of low abundant proteins, presence of interfering compounds like salts, or none of the above. Yes. Which one should be the answer? C. Yes. Presence of interfering salts. In a gel, there are two proteins with same isoelectric point, but they differ in the molecular weight. Okay, they are having same isoelectric point. Means in the first dimension, they will be at the same location because they are having same isoelectric point, but they differ in molecular weight. So what will be the position of these protein spots on the gel with respect to each other? At the same position, protein spots on the gel with respect to each other. On at same position, they will be overlapped, adjacent to each other, vertical to each other. Yes. You can run two protein samples in two separate 2D gels. So you have run that. Then now you want to study and compare the differential express proteins. Which of the following software can be used? Okay, we didn't talk about the software name as such, but if you want to compare differentially expressed proteins, so there is one software which is called as Image Master Platinum IMP7. This is something which is used for studying the differentially expressed proteins. Which of the following is a correct sequence of events followed followed for serum proteomics? So just look at it. So obviously, if you see, we need to do initial step and we'll quantify later. So it can be either the first answer or third answer. So what do you think? So as I just give one of the hints, so quantification will be obviously the last step, right? Then there should be some initial steps. So which one will you generally do? First, you will need to lyse the cell. For lysing the cell, do we use depletion or you need to use sonication? So which one should be the answer for this question? Yes. Correct sequence for serum proteomics study. As I already gave a hint, it final step will be quantification. So it will be either first answer or third answer. Which one, A or C? Yes. You all are confused? So first you will lyse the cell, right? You will sonicate the cells first, then depletion, then protein precipitation, and then quantification. So it's sonication, depletion, protein precipitation, and quantification. Which of the following statement is not correct for 2D gel 2D rehydration process? So rehydration process, which one is not correct? Salts removed in this process, mineral oil is added to prevent strips from drying, activates the charge group on IPT strips. This is easier to answer, I think. Yeah. Are you all there, Lekha and yeah, option A, that is something which is not correct. Is this still correct? It doesn't activate the charge group on the IPG strips. Option A, B. I think this answer is wrong here. Activates the charge. The answer should be A because these are the two things which happens while we do rehydration process. Salts are removed in this process. That is something which is not correct. These two is something which is correct. But in this specific PDF, I think this is something which is written, which is a wrong answer here. The answer should be salts are removed, which is not correct. Yeah, in two dimensional, the component of the gel which maintains the pH gradient are called. This is a general knowledge question, though although we didn't discuss, but which is the component which maintains pH gradient? They are called as, if you all can answer. Timid? No, not timid though. It's ampholites. Timid is a initiator of cross-linking, so it is nothing related to pH gradient. Polytrimide is the something which gets cross-linked. You need to get confused between either A or C, as Lekha was confused, but the answer is ampholites. In two-dimensional, second dimensional electrophoresis is done at dash degree angle relative to first dimension. The first dimension is like this. Second dimension is the first dimension is like this. Basically, you just load the sample and run it on a gel. Then the second dimension is like this. So what is the angle which is happening? It's 90. Yeah. So in 2D gel electrophoresis, oh sorry, where am I? So in 2D gel analysis software, which parameter is used to compare gels on spot by spot? It's the question itself is answering you, giving you the answer. So which is spot by spot basis analysis? It's spot analysis. Yeah. Which of the following stain dye is not compatible with mass spec? Which of it is not compatible with mass spec? Massive Cypro Silver Duel. There was a sheet in the final comparing all of these techniques. So the answer is silver staining, which is not compatible with mass spec. So in silver staining, the reaction is stopped by adding. We did discuss about this. The reaction needs to be stopped by adding ethanol, acetic acid, ethanol, water, acetic acid. Which of the following statement is correct about isoelectric focusing? Proteins are resolved on the basis of their is correct. Involves the use of immobilized pH gradient, presence of salt interference in IVF process. When this is easy to answer, all of the above. Yes. Which of the following statement is true for image quant TL software? Oh, so we haven't dealt with software as such. So still you can just guess. Uh, it can remove the background by background subtraction. It can be, so it is image quant TL. It can be used for quantitative proteomics. So quant, it can employ standards for molecular weight studies. The answer is all of the above. So I think these questions are about the software. We are not looking at. So this software, so you can, there was a lecture basically by one of the sir in the third or fourth lecture of this week. You can go through that specific like final, maybe final 10 or 15 minutes where sir discussed about one of the software. If I'm not wrong, I think this is the software which sir discussed, Image Quant TL. So you can go through that once, and he actually opened the software and talked about the software, what all that software can be used for. So I haven't discussed this in the lecture, but you can go through that. I think it is on the third or fourth part of the week 3 lecture, and you can go through that software. I am not having that software here, so I'm not covered them as such. But you can go through that, and it will help you to answer these questions. So in that software, which method is preferred for 1D gel analysis? Automatic, stepwise, or AB? It's A and B. Which of the following software analysis can be performed by this software? So it can do colony, it can do Dij, cannot be performed. Sorry, that's what I was confused because it can do 1D gel analysis. So it is cannot be performed. Is Dij analysis something which it cannot be used to perform? And you run a SDS page gel and wanted to analyze differentially expressed proteins from serum samples, and you were doing analysis using the same software and wrongly selected one of the following parameter. So which did not give you desired results? What is that option in the software? Fix the bandwidth, automatic bandwidth, automatically band detection. So it is fixing the bandwidth. So once you do that, because of some wrongly selected one, it gets corrected. So last four questions were about the software. If you go through this last 10 minutes, four questions as well. Yeah, so that's it from my side for today's lecture. I'll upload this specific as well as the PPT. If you all are having any doubts, you all can ask me. We are still having around 5 minutes or so, or else we can discuss. Great. Yeah. Anything else? Anyone? Aditi, Lekha? I don't remember, but where are you all from? Like you are currently doing what? Lekha and Aditi? Masters, bachelors, and where are you all from? Yes. I just entered in BTech, Biotech. Oh, okay, you're from Coimbatore. Nice. And what about Aditi? If you're there. Okay. Uh, thank you for attending. We'll meet again. I think that is the last lecture. If I get a chance, maybe I can take one more lecture before your exam as well, combining all the four weeks lecture together. I'm planning to do that, but if I don't know if I will get that opportunity to do it. Next week is possibly the last lecture, or else if I get a chance to just give an overview of all 4 weeks lecture, I might take one more extra lecture. But if not that, then the next week lectures will be the last one. And then after that, you may have to appear for your exams on in the month of September, if I'm not wrong. The exam questions can be solved if we follow the weekly lecture materials only. Yeah, you can. Ideally, that's how the question should be. You, but you need to understand those weekly lectures in a good detail so that if there is questions like थोड़ा घुमा के वगैरह questions पूछे तो भी still you need to understand that कि it is related to that only and you will be able to answer. Yeah, it will be related. It will not be something which is out of the week, all week 4 lectures, all weeks lectures. Yeah, it will be related to that only. Okay, thank you for attending.